In this lecture, we turn our attention toward the special senses that are going to detect light and chemical information from the world. Chemical sensation is going to be through the mouth and the nose, so it'll be taste and olfaction. These are going to work together to create flavor. Optical sensation is going to take place in the retina. There's a very small component of this where we have direct light sensation from retinal ganglion cells, but your typical visual circuit is going to involve detection of light by photoreceptors, stimulation of bipolar cells, then stimulation of retinal ganglion cells, which project back to the lateral geniculate nucleus of the thalamus and eventually hit the visual cortex to create visual perception. We're going to start off with taste. We're going to have a mix of G protein coupled and ionotropic receptors here to detect tastes. And there are 5 of these tastes. We have 5 different taste cells that all express one type of receptor to detect one type of taste. Now within each taste bud, you have all different types of taste cells, so there's no taste map on the tongue. Every taste bud is going to have each of the 5 different types of taste cells. The only difference between the front and the back of your tongue is which cranial nerve innervates it, not which tastes we're going to pick up. So each one of those taste buds is going to have 50 to 100 different taste cells, so every taste will be represented. The taste bud is going to have a little pore there that allows chemicals to diffuse in and interact with the microvilli on the taste cells. Different receptors in the microvilli are then going to pick up tastants. The taste cell is going to spit out some ATP to stimulate the gustatory nerve. This is going to be a combination of the facial and glossopharyngeal nerve. So we're going to have a taste cell, and there will be a few of these in the taste bud. So we'll put a couple on here. These are going to be innervated by fibers of cranial nerves 7 and 9. The neurotransmitter here that we're going to be releasing is ATP, and that's going to act on purinergic receptors to stimulate our cranial nerve there. This will head back into the gustatory nucleus and the nucleus of the solitary tract, and that will eventually hit the thalamus, work its way up to the cortex to create our perception of taste. We know that ATP is important here because if you knock out P2X2 receptors, in this case the chordotympani nerve, so this is a component of the facial nerve here, no more response to any tastants, although the nerve still responds to temperature and touch. So ATP is the neurotransmitter here that's going to go from taste cell to cranial nerve. Stimulation of the cranial nerve then leads to a change in activity in the central nervous system. We got 5 tastes. We're going to walk through them one at a time. So, umami would be the savory taste, and when we're talking about humans, we're talking mostly about glutamate. So MSG is going to stimulate umami. Sweet is, of course, stimulated by saccharides. Now there's some similarities here between these two. So they're going to have G protein coupled receptors in the taste receptor 1 family. So the taste receptor 1 family, here we're going to have a couple different types. So we're going to form dimers. So dimers of the 1 and 3 subunits of taste receptor 1 is going to create the umami. We're going to have a different combination here to detect sweet. But same idea here, G protein coupled receptors. So, these are going to be either metabotropic glutamate receptors or metabotropic sugar receptors. And we're going to have heterodimers here. This chart's just going to come up a few times to show you what different knockouts are going to do to the detection of different tastes. So whenever you knock out T1R1, for example, you're going to lose umami. T1R2, you're going to lose sweet. T1R3, you're going to lose umami and sweet because they form dimers there. T2 taste receptors are going to detect bitters in this case. So, these are all G protein coupled receptors. So the T2 receptor family is going to create the bitter. Now, in each case, our taste receptor family, these are going to be GQ coupled. So these are, of course, going to increase intracellular calcium, just like they did in Lecture 9, and that's going to stimulate the release of ATP in taste cells. The elevation in calcium is going to stimulate TRP channels that are going to further drive depolarization, and then stimulate voltage-gated calcium channels. The mechanism is shown here in this cartoon. So our G protein coupled receptors are going to pick up umami, sweet, and bitter. The sour taste, so that's our fourth taste here, so umami, sweet, bitter. Sour is going to be detected with TRP channels. So now we're going to move on to ionotropic receptors. So we use GPCRs for these. We're going to use TRP channels. This will be PKD1 L3 for what that's worth, and PKD2 L1. So these are just components of TRP channels that are expressed to detect bitter. Sour. Sour in this case is going to be acid. So things like lemon juice, things that are acidic. These are going to stimulate TRP channels, kind of like they did when we talked about nociception. So acid sensing ion channels is what these are. So the TRP channels are going to depolarize taste cells and lead to then the release of ATP. Here we can see colocalization of these two TRP channel components in taste cells. So if you just look in C, you'll notice the green and the red, these two subunits, green and red, colocalize, and we see quite a bit of yellow. The bottom is just showing you that in taste tissue we do see co-expression of these TRP channel subunits. We of course see them in the, I believe, the testes as well. Yeah, you see everything in the testes, so that makes sense. We don't do any tasting with those though, in case that was a thought. Application of acid, that's what we're looking at here, is going to cause a robust inward current. In other words, we depolarize. And that's what we're seeing in these data here. When we depolarize, we spit out ATP and stimulate the gustatory nerve that's gone. Sodium detection, very simple. Sodium channels, just direct depolarization by sodium. Things taste salty because sodium is present. Sodium flows in through sodium channels and causes depolarization. Pretty simple mechanism there. It's not so much about the tastant as it is the taste cell that's stimulated. So this brings us back to our labeled lines. What we're looking at here in these data are how much a mouse likes water that contains this novel bitter molecule. Mice can't detect it, so the wild type control there, you'll notice there's no preference. So they're given two types of water, with the bitterant and without. And we're looking on the x-axis with an increasing concentration of this bitterant. If you don't have the bitter receptor for it, you don't detect it. When they put the novel bitter receptor into bitter cells, those that express the T2 taste receptor family, they see a decrease in preference because it's bitter. Mice don't like bitter water, but they like sweet water. Now, even though they haven't added any sugar, they still added the bitterant. They put the bitter receptor into sweet taste cells. So we had two options here that we could express in. Either sweet or bitter. And so we have some novel taste molecule here, TM taste molecule. Now normally this doesn't bind to either of these in mice. If we put our bitter receptor here, then we sense bitter. Mice don't like it. On the other hand, if you put the same receptor into sweet cells, the same bitter component now tastes sweet. This illustrates labeled lines. Stimulate this taste cell, no matter what does it, we perceive sweet and we like the water. Stimulate this bitter taste cell, no matter what's stimulating it, we perceive it as bitter. We like sweet, we don't like bitter. And we can see the data there in their preferences. Olfaction is going to require a much larger number of receptors, but they're all going to be G protein coupled receptors. So that's kind of nice. We just have GPCRs. Now the olfactory sensory neurons are going to dangle their dendrites down there into our nasal cavity to sense what's in the air that we're sniffing. And it smells kind of like markers in here right now. And that's because little molecules that diffuse off of the marker go in and bind to G protein coupled receptors. They bind to odorant receptors. Now these odorant receptors comprise a large number of G protein coupled receptors. That's what olfaction's all about. This is an enormous family with at least 1,000 different types of odorant receptors, and that allows us to detect so many different smells. Whether they be pleasant or otherwise, we have a receptor for it. Now, some olfactory sensory neurons are going to detect more than one odor, but that's not because they express more than one odorant receptor. They still only express one, but some odorant receptors can bind to multiple different odorants, while others are going to be more specialist and bind to only one odorant. But every olfactory sensory neuron expresses only one odorant receptor. Only one type. Odorant receptors are GS coupled. Now they call it golf, or G olfactory alpha subunits, but they're just GS. They still stimulate adenylyl cyclase, they still increase cyclic AMP levels, and those still stimulate cyclic nucleotide gated cation channels. Nothing simple there. That drives, nothing complicated there. That drives depolarization through cation influx, stimulating voltage-gated calcium channels and causing glutamate release. That glutamate release in the olfactory bulbs is going to then stimulate secondary afferents that'll project back directly to the cortex, it turns out, to help create some emotional response to that olfactory input. Higher order neurons are going to actually create our perception of this odor. Now the projection pattern of olfactory sensory neurons is pretty unique. They're going to all hook up together in the olfactory bulbs based on which odorant receptor they express. So in my nasal cavity here, we're dangling our dendrites down to sense odor. Let's say these neurons here have the same odorant receptor. So air comes in and we smell it through GPCRs on our dendrites here for olfactory sensory neurons. If they express the same odorant receptor, their axons are going to meet up in the olfactory bulbs, in the glomeruli. So a glomerulus is just a region where axons collect. Axons from primary olfactory sensory neurons all meet up in different glomeruli. Now, if these two express the same odorant receptor, they'll meet up in a different glomerulus. We haven't figured out any sort of mapping here, but it is most definitely the case that olfactory sensory neurons meet up in glomeruli. We don't know how, but certainly the odorant receptor plays a critical role. Because when you delete an odorant receptor, you see the loss of that glomerulus. So if you'll look in the data here, on top they've deleted an odorant receptor. On the bottom they've kept it in place, and in both cases they're expressing LAC-Z so we can see the axons. You'll notice the emergence of a glomerulus over time in the second row, but in the top row, nothing. Those olfactory sensory neurons lack their odorant receptor, therefore they're not going to be functional, they won't form a glomerulus, and they'll eventually die. In all likelihood, this has to do with the expression of different surface proteins that allow them to differently follow guidance molecules or stick to each other. We don't really know how. We do know that they meet up in the olfactory bulbs at glomeruli. From there, we're going to go back into the cortex, and something magical is going to happen. Now, the detection of light is going to take place in the retina. So, the retina is going to have a couple of holes in it, one in the front to let light in, and then a hole in the back to let nerve fibers and blood vessels come in and out of the retina. So we're going to be passing light through the cornea, through the pupil, through the lens, and the lens is going to help focus the light on the retina. The retina is where the magic happens. We got 3 layers in the retina. There's the outer nuclear layer, there's the inner nuclear layer, and there's the ganglion cell layer. Now, of course, we're all aware by this point that our retina is inverted. In other words, the ganglion cell layer is on top, the outer nuclear layer is on the bottom, and the inner nuclear layer is in between. So when you look at that eyeball there, we're actually going to have our retinal ganglion cells on top. Then we're going to have that inner nuclear layer here that contains our bipolar cells, and the outer nuclear layer with the photoreceptors. So light has to pass through these 2 layers here before it hits the outer nuclear layer. This inverted arrangement also means that the axons from our retinal ganglion cell are going to pierce a component of the outer nuclear layer. Thus it will be devoid of photoreceptors, and we will have a blind spot. Now, in that outer nuclear layer, we have 2 types of photoreceptors. We got rods and cones. Rods aren't sensitive to different types of wavelengths of light, so they will be color neutral. Cones are going to detect broad wavelengths of light, so they'll start to tease apart different colors. The basic circuit that we all need to know is there's some kind of photoreceptor, and they're usually drawn like this. These are going to communicate with bipolar cells. These then stimulate retinal ganglion cells. These are the neurons that then project back through the retina and up to the thalamus. This is the basic circuit. Now we also have horizontal cells that provide some inhibitory output to neighboring photoreceptors. And we have a couple types of bipolar cells. There are amacrine cells that are going to have modulatory functions here as well. They're fairly complicated, so we're going to leave them out of this and try to stick with the basic circuit here. So how do we detect light? We're going to detect it, of course, back here in the outer nuclear layer. Stimulation of the retinal ganglion cells is going to be produced by this stepwise movement from photoreceptor to bipolar cell to retinal ganglion cell. That piercing of the retina then creates the blind spot, like I said before. Alright, how do we actually detect light? Well, it's of course with pigments. Pigments are pigmented because they absorb light. So my jacket is blue because it absorbs all kinds of colors but blue. So blue bounces off and hits the camera, and that blue is then projected out for you to detect. So there's 2 parts to photopigment. There's the retinol, which will actually absorb the light, and then there's the opsin, the G protein coupled receptor that's going to actually do something. Now, different types of opsins are going to have different effects on retinol, and they're going to change the color of that retinol through their interactions. we got three types: L-opsin, M-opsin, S-opsin. Long, medium, and short wavelength. So you'll see they detect more of the kind of reds, greens, blues. Not totally true, but that's how we're going to think about them. Rods have a color-neutral rhodopsin, very broad spectrum, so it's going to just detect light. It doesn't care much about color. When light hits photoreceptors, that's going to lead to hyperpolarization. The way that it's going to do that is by stimulating transducin. So, this is a new alpha subunit. What transducin is going to do is activate a phosphodiesterase. So, light is going to stimulate opsin. Opsin stimulates transducin. Transducin stimulates phosphodiesterases. The phosphodiesterase is going to break down cyclic nucleotides. These cyclic nucleotides, of course, stimulate cyclic nucleotide-gated cation channels. So when light hits, we break down our cyclic nucleotides and we decrease conductance through our cyclic nucleotide-gated cation channels, causing hyperpolarization. Now there's a constant efflux of neurotransmitter out of our photoreceptors. So they have those ribbon synapses just like our hair cells in the inner ear. So they're always spitting out glutamate. That's what we're releasing here. Glutamate. That's going to stimulate horizontal cells and bipolar cells. So when we have light, we have less glutamate. We're releasing less glutamate as a result. That's going to affect bipolar cells depending on which receptor they express. That's also going to affect horizontal cells. And in doing so, that will affect neighboring photoreceptors. We'll get to that in just a bit. Now, the distribution in the retina of rods and cones is not uniform. So if this is our fovea here, the center of our visual field, this is going to be enriched in cones. So we have nice color vision wherever we're looking. As you move away from the fovea into the periphery of the retina, you see an increase in rods. That means the periphery of our retina is relatively colorblind. That's what these data over here are showing us. Cones are almost exclusively in the fovea, rods more in the periphery. Rods are more sensitive to dim light, and that's going to allow us to have a better detection of dim light in the periphery, and that's why you look away from a star in order to see it. Now, the effects of this change in glutamate is going to depend on which glutamate receptors do we have. So, if you have ionotropic glutamate receptors, these normally depolarize you, so the decrease in glutamate will turn you off. Off bipolar cells have AMPA receptors. mGlu-Rs, on the other hand, are going to be expressed on bipolar cells. These mGlu-Rs do the same thing that goes on here. They're going to stimulate a phosphodiesterase that breaks down cyclic GMP, so these normally turn us off. When we lose glutamate, the bipolar cell gets turned on. So depending on which bipolar cell is here, light can either lead to an increase or decrease in retinal ganglion cell activity, depending on whether there's an on or off bipolar cell there. If it's on, decreasing glutamate causes an increase in glutamate release here and thus stimulation of the retinal ganglion cells. Off gets turned off by the loss of glutamate, thus we have less stimulation of retinal ganglion cells and reduced output. And here you can just see the cartoon. Ionotropic receptors or metabotropic receptors. The horizontal cells are going to help with edge detection. And what they're going to do is allow areas where we have transition between light and dark affect each other differently. And it's going to amplify that difference. So, when we decrease glutamate output here, what that does is decrease the activity of horizontal cells. Now these are inhibitory. So normally these are going to cause hyperpolarization here. So the hyperpolarization of this photoreceptor here leads to depolarization of its neighbor because of reduced GABAergic input from horizontal cells. And this is going to help with contrast detection. It's also going to create polar opposite effects depending on whether the light is in the center of this retinal ganglion cell's receptive field or in the periphery. Think about what would happen if we shone the light here, not here. We're going to hyperpolarize this photoreceptor. That's going to lead to a decrease in GABA release here in the surrounding photoreceptors. They're going to depolarize and we'll get an increase in glutamate, making this section appear darker than it did before and getting the opposite effect here on this retinal ganglion cell. So horizontal cells are going to help with two things: detecting edges, and they're going to create that on-off center surround that we see in retinal ganglion cells. Now there are some special retinal ganglion cells here that bypass all this crap and have photopigments themselves. So some of them are going to express melanopsin. Melanopsin, just like in the photoreceptors and the mGluRs, is going to stimulate transducin. Same idea here. So they're going to be sensitive to light levels all by their own. So these are the retinal ganglion cells that project back and help create our circadian rhythms. Because you don't need any of this stuff. You don't need photoreceptors and bipolar cells. So if you'll look in the images, that's a cross-section of the retina. On the left, that's good old wild type. You'll notice the pigments. So the photoreceptors there have been destroyed in part D. So here's a mutant that has retinal degeneration, and that's caused by the loss of photoreceptors, not retinal ganglion cells though. In the bottom, you can see circadian entrainment based on shining different blasts of light. So different light intensity is going to create different degrees of circadian entrainment. The brighter the light, the greater the entrainment. There's really no difference between wild type and these retinal degeneration models if they affect just photoreceptors. So these have nothing to do with circadian rhythms. We bypass it with some special retinal ganglion cells. These are sensitive to blue light, and that's why the night mode for screens is going to look mostly yellow. They get rid of the blue light. Now that center surround inhibition that we see with retinal ganglion cells, where we get stimulation in the center but inhibition in the surround, is caused by horizontal cells. Make sure you can walk through this basic diagram here. So if we have an on bipolar cell here, shining light onto this photoreceptor, so if we think about this, here's the center, here's the surround for this retinal ganglion cell. Light in the center stimulates the photoreceptor, that then stimulates the on bipolar cell and stimulates the retinal ganglion cell. So light in the center leads to an increase in activity. Light in the periphery, though, is going to lead to a decrease in activity, because if we shine here, we hyperpolarize this photoreceptor. We don't hyperpolarize this one. Instead, we depolarize it because of reduced GABAergic input. We apply more glutamate onto this bipolar cell. That stimulates mGluRs. Normally these inhibit the bipolar cell, so it releases less glutamate on a retinal ganglion cell. We see a decrease in output. Now, there's different types of retinal ganglion cells that can be distinguished based on their size and their projection pattern. So we got P cells and M cells. P cells are going to project to the parvocellular layer. M cells are going to project to the magnocellular layer. So P cells, they're going to have much smaller receptive fields. These are going to be those cones in the fovea, so they're going to be sensitive to color. The M cells are going to be a lot more sensitive to dim light, so they're going to be found a little more in the periphery. They're going to have larger receptive fields, They're going to be a little more sensitive to movement because of their rapid desensitization. So they're there for detecting dim light in the periphery and things that are moving. They're going to hit, if they're P cells, the parvocellular layers. So 3, 4, 5, and 6. And then the first two layers of the lateral geniculate nucleus, the magnocellular layers, are going to be hit by M cells. So those P cells are color sensitive because they're hooked up to cones. So in the fovea, where we have all of our cones, that's where we're collecting our P cells as well. Those retinal ganglion cells are much smaller, so we can pack a bunch of them into a small space. And by having an increase in the density of retinal ganglion cells, we have an increase in our visual acuity right there in the fovea. That's why we look at stuff when we want to look at it, and not away from it. So if you want to read some text, you look right at it, not away. You have the best visual acuity there. In the retina, there's probably a little bit of color detection going on, but not much. Because for the most part, we have one-to-one coupling. But there's a little bit of color opponency. This is more true in the thalamus, but we'll go over it here. So for color opponency, we're comparing the amount of input that we have from the long, medium, and short opsons. So these will be the different types of cones. If it's red, we're going to see a lot more stimulation of long. If it's yellow, we'll see stimulation of L and M. If it's green, we'll see more stimulation of M. Again, this is just relatively speaking. We'll see more activity here. If it's blue, we'll see S. If it's white light, we're going to hit them all. Now remember, we have on and off bipolar cells, so we can do a little bit of comparison here. Light doesn't have to turn us on, it can turn us off. Mostly we're going to be doing this comparison in the thalamus. But we can distinguish kind of broad colors from one another. So if it's white light, I'm going to be turned on by any of these as long as there's an on bipolar cell between them. So if we wanted to make a neuron, a retinal ganglion cell, let's say, sensitive to yellow light, and only yellow. We don't want white to turn us on as much. We only want yellow. What we're going to want is this pattern of bipolar cell input. On between our L and M cones and off between our S. So as long as there is red and green light, that makes yellow, but no blue, because that would make us white, will be turned on. On the other hand, let's say we only want to be sensitive to green. That's not too tough. We just got to make sure we don't get excited by red light anymore. If we wanted to be sensitive to blue, we'd put an on here and an off here. So depending on the retinal circuitry between photoreceptor and retinal ganglion cell, or a lot more likely photoreceptor and neuron in the lateral geniculate nucleus, we can start to tease apart broad colors in our vision. And what these data here are showing us is just the different sizes in our P cells and M cells. Now, it's very unfortunate here that we have the names midget cells and parasol cells, because M, you might think would be midget, but no, it's parasol. So the M cells, remember, are large, magnocellular. So the parasol cells, I guess it kind of looks like an umbrella, these have very large receptive fields. So if you look in the top, this is a cross-section through the retina, and we're looking at retinal ganglion cells. In the fovea, we're going to see very small dendritic arbors. B, that's showing us a top-down view of their dendrites. So very small dendritic arbors in our P cells. Very large dendritic arbors in the M cells, or in the parasol cells. And that's what makes them more sensitive to dim light. They're going to be summing all of that glutamate input from bipolar cells in that entire dendritic arbor. not just a little area. That's why we're going to need very bright light for us to see it and make sense of the color. Not true for just dim light in the periphery. From the retina, we go to the thalamus. From the thalamus, we go to the visual cortex. Then we're going to have some dorsal and ventral streams that we're not going to talk about in this class. That'll help us figure out where and what, respectively, we're looking at. And that's it for sensation. That's how we detect environmental stimuli, whether it be pressure or something a little more special, like light and chemicals. If you have any questions on the material, fill out the questions box. I'll see you later.